All right, so in this section, we're going to talk about gene expression, or that conversion from DNA into proteins. So the term of which we're going to use, gene expression. So protein synthesis is going to occur in two steps, transcription and translation. Transcription is the first step, involves transcribing the original DNA message. So during the process of gene expression or protein synthesis, transcription is going to copy that DNA into RNA. Translation is going to turn that RNA into proteins. Proteins are comprised of subunits called amino acids. There are 20 different amino acids, and proteins are going to differ in their number and order of their amino acids. Just like you can write a book with only 26 letters, and the combination of letters, the order of the letters, the length of the sentences, the length of the words, all of that contributes to the book that you are creating. You can kind of think of the human genome as a book. Proteins are going to be made up of only 20 different amino acids, but you can put them together in any sort of combination. You can put them together in any order. You can put them together at any length to create whatever sort of proteins that you want to create. The sequence of amino acids in a protein is going to ultimately determine its particular shape. And as we've already mentioned, when we talked about enzymes, the shape and the shape of the protein is absolutely integral to its function. So structure is going to be hugely important, and structure is going to contribute to its overall function. So here is just an overview of the process. Transcription is that first step. The sequence of nucleotides in DNA is going to be copied into a sequence of nucleotides in the mRNA. In other words, that transcript. So here's our original DNA sequence. We're going to copy that down into an mRNA transcript. We're then going to translate. We're going to translate that mRNA transcript into amino acids. So that mRNA is going to be translated into a sequence of amino acids. Now, here's the interesting thing. Translation from mRNA into amino acid sequence is going to work much like translation from one language into another. When we are translating Spanish into English or English into Spanish, we don't translate one letter at a time. It would make no sense. We use the same letters in Spanish as we use in English. Instead, when we are translating, we translate word for word. We translate one word and we turn it into the equivalent. That's going to happen the same way in mRNA. In the process of translation, when we translate from mRNA into polypeptides, we are going to do a word-for-word translation. The words, the so-called words, are what we refer to as codons. So codons are going to be a 3-base sequence in mRNA. Each 3-base sequence in mRNA is going to represent a specific amino acid. It's kind of like how a three-letter word is going to represent, like it's 3 letters, but it still represents a whole word, like the word cat, C-A-T. Cat is it's a whole word. It means something. It is an animal, but it's made up out of those 3 letters. So, yes, so we refer to a 3-base sequence in amino acid, a 3-base sequence of RNA as a codon. Each codon represents a specific amino acid. There are 64 possible codons, 61 of which correspond to a specific amino acids. The remaining are sort of punctuation codons. They're going to be stop codons or start codons. They're going to let us know when the sentence begins or ends. One of those codons, AUG, specifically stands for the amino acid methionine. Methionine signals for the beginning of the polypeptide. Most amino acids have more than one codon. So what we're going to see in our table on the next page is that there is quite a bit of redundancy in the genetic code. This is to help protect us against mutations. So just for an example, you can see that here the codon UAA codes for the amino acid tyrosine. Also the code UAC also codes for tyrosine. So just in case we had some sort of mutation and that U was mistranscribed as a C, we're still going to get the correct amino acid sequence. So there's quite a bit of redundancy built into this code. It's going to help guard against or protect us against mutations. Now, the way you're going to use this genetic code, because you will be using it, it's sort of like your translation dictionary. It's going to translate from nucleic acids to amino acids, is you're going to start off by looking at the first base. So let's say we wanted to translate the codon AUA. We're going to look at the first base is an A. So we're going to go down here. Okay, this is the area where first bases are an A. The second base is a U. So we're going to scroll across the top and we're going to look for where the second base is a U. That's going to be in this column. So we found the A's, U, and then the third base is also an A. So that's going to be down here in this row. So the intersection point, or where all three of these intersect, is going to be right here. First base A, second base U, third base A. AUA is isoleucine. If we wanted to, actually, I'm going to let you do this. So take a moment and see if you can tell me what the codon is for. See, do we have codons over here? Yes. Here we have some more codons. See if you can determine or translate the codon AGC. So that's going to be my next challenge for you. See if you can use this table to translate AGC into its corresponding amino acid. So we're going to talk just briefly about the process of transcription. Like we already said, this is going to be where we copy down our DNA message into RNA format. During transcription, a segment of that DNA is going to serve as a template for the production of an RNA molecule. So what we're going to do is we're going to unwind a very small section of the DNA. Now, we're not going to unwind the entire strand of DNA. Like I said, we're not going to sit down. We're not going to read the entire book cover to cover. We're going to just unwind that one section of DNA that we need, and we're going to unwind it just long enough for our RNA to match, to do that complementary base pairing with that section. So we're going to use RNA polymerase. We're going to work from a 3' to a 5' end, and we're going to just read a small sequence of DNA, and we're going to copy that down using our nucleotide base pairing rules to make our RNA strand. So in this case, if you look at this region right here, our DNA strand is CGT. We are going to use our complementary base pairing rules to fill in the RNA sequence. C must bind to G, G must bind to C, and T must bind to A. So if our DNA sequence is CGT, our corresponding mRNA sequence will be GCA. Please let me know if you're having any questions or concerns with DNA transcription. We will be covering this pretty extensively during our live discussion meetings, but this is going to be a very important thing for you to be comfortable with, as you will see this on homework and tests, where you will be given a short section of DNA and you'll be asked to give the complementary mRNA sequence. Once we have copied down our mRNA transcript, we still have to kind of process it. You can kind of think, I don't know about you, but I have found that some of the best ways for me to take notes or to study is repetition. So a lot of times when I take notes in class or when I take notes in my textbook, I copy down large portions of information that I think is useful. But when I want to actually study that information, when I want to actually put that information to good use, I will just copy it down again as a bullet points form. I will only take the most relevant information or the information I don't know. One thing I found is a very helpful way for me to study is to make flashcards of everything. And I'll go through the flashcards, and any question I didn't know right off the top of my head goes in one pile. Anything I could answer without even thinking about it goes into another pile. And then I just focus on the ones that needed a second. And so then I'll go through the whole stack of flashcards again, the ones that needed, the pile of those that needed my help. And then I'll go through again. Anything I was able to answer the second time around pretty easily, that'll go into the discard pile. Anything that still needs work, I'll keep working on. So my pile of information is going to get a little bit smaller each time. As I'm copying down those notes, they're going to become more and more efficient and more useful for note-taking and studying purposes. mRNA is going to be processed in a similar way. So what happens is that we take our new primary mRNA strand and we then have to mature it. We're going to have to process that before we can leave the DNA, before we can leave the nucleus. An RNA sequence is going to be made up out of introns and exons. Introns are segments of DNA that interrupt genes and are they're not really part of the gene. They don't code for a functional protein. Exons are going to be portions of the genes that are expressed. So you can kind of think of introns as in the way and exons as the expressed portions of the gene. So the primary mRNA contains both exons and introns. This is where we just copy down all of the information straight out of the DNA. Then what we're going to do is process that information. We're going to put a 5' cap on one end and a long tail on the other end. This poly A tail, it says poly A, that poly A tail just means a lot of adenines. We're going to take a lot of adenines, and we're going to tack them onto the end of this. This long poly A tail helps feed the mRNA sequence out of that tiny little nuclear pore. It's kind of like a needle and a thread. It helps, that stiff region helps feed it out of the nuclear pore. It also is going to prevent degradation. If we were to experience a degrading enzyme, an enzyme that's going to start breaking down that mRNA once it's in the nucleus, once it's in the cytoplasm, it's going to start on the 3' end, and it's going to start slowly chopping those poly A's off. Since we have a huge long sequence of adenines, and usually, when I say a lot, there's usually like probably between 200 and 500 adenines that are attached to the end. Those are going to slowly be broken off, but the entire message is going to remain intact. So we're going to add a protective cap to one end that says, hey, this is the beginning of the sentence. You need to start here. We're going to add a tail to the other end that's going to help feed it out of the nuclear pore and prevent it from degrading. And then we're going to splice the mRNA. What's going to happen is we're going to use these little proteins called spliceosomes, and they are going to remove the intron portions and keep only the exons that are going to be expressed. This spliceosome is composed of both RNA and protein. The RNA portion, not the protein, is going to be functioning as the enzyme. So it's sometimes referred to as a ribozyme. Now here's the interesting thing. There is also the potential for alternate mRNA splicing. When I was younger, when I was first taught about mRNA, and way back in high school, we were taught that these introns were always referred to as junk DNA. The idea being that they don't code for anything, and so they're just in the way and they're kind of pointless. However, there has been a lot of research that has been done in the last few years about spliceosomes and RNA splicing. And what they have found is that you can actually create multiple proteins from a single segment of DNA if you splice it differently. So it's possible that maybe this time the message we want to code for our protein is here, here, and here, but maybe next time we're going to splice it differently. We could splice and retain this portion and this portion and this portion. Or maybe we want to keep this portion and this portion. And in those ways, we can actually code for multiple proteins with only one sequence of DNA. And what they have found that is in sort of higher complex creatures, the difference between like the DNA of a human versus the DNA of a flower, it's not so much about the number of chromosomes. A lot of plants have so many more chromosomes than humans. They'll have, upwards of 100 chromosomes. We only have 23 different chromosomes. So if you were to simply look at the amount of DNA that humans have, you would think humans are not really all that complex. We don't have that much DNA. But what we have compared to every other creature on Earth is we have so many more spliceosomes and ribozymes. And what we are finding is that the complexity of the organism often comes down to the efficiency of their DNA sequence. Not how much DNA they have, but how many different proteins can be created from that single DNA sequence. So anyway, that was sort of my small little tangent. We will pick up with translation in our next video.